Okay. So, because we still have a fair number of things to cover, and we've made it to slide 17 of 54, we should probably get going again. So, the idea for kind of the reproducible analysis is that, all of the analyses that we do are sort of scripts or macros or workflows in the case of time, that we want to be able to perform the same analysis many, many times very easily and see what the sensitivity different parameters are. And so if you're using a tool like MATLAB, it might look something like this, where you have, some code with parameters defined above that you then run as a script. If you're using an ImageJ, you can then run things like ImageJ macros very easily from the command line by doing something like that. If you're using R, you can run it like that. For 9, it's a little bit more complicated, but it's more or less the same idea. And what this means is that you can basically run your whole analysis hands-free, so that everything you need to run the analysis is kind of contained in that line, and you don't have to manually click or interact with anything for it to run. This is also kind of the idea behind Kaggle. And so if you've looked at all of the kernels that are available inside of the Kaggle platform, all of them are entirely self-contained. And so you know exactly what versions of what libraries are installed and available, and they run on their machine with this pre-configured setup and the data to do your analysis and show the results. And you know that anyone taking that machine with this script will be able to get exactly the same answer. And so you can be very confident that your results are meaningful, and you give the ability for anyone else to kind of look at your analysis and decide if that's what they want to do or if that makes sense or not. And that's one of the things that's very important to have, is that you are able to have other people repeat your results. And this is why you write scientific papers with a very long detailed methods section, so that someone else would be able to read this and attempt to do what you did. So if we go for the reproducibility, how can we go about measuring this? And so one of the easiest tools we have for this is called intra-class correlation coefficient. And so if we go back to the iris data set that we showed before, we can look at this variable called sepal width. And we can take it for each species that we have. So Setosa, versicolor, virginica. And we can show sepal width plotted as sort of a box plot or individual points for each one of these different species. And so one of the things that we see here is there's actually a fairly low group similarity. So when we look at the different groups that we have here, the variation within the groups is very large. And how are we able to say that it's very large? I guess I framed that question a little bit wrong. But when we look at this group and we say that this has low group similarity, and we look at this and say this has high group similarity, what does that mean? Yeah. And so when we see these, there's a lot of overlap between them. And what that means is that the variation within a group is larger than the variation between groups, or is on the same order. And that means that they are similar, or they have low group similarity. And when the variation within a group is smaller than the variation between groups, we say that it's a high group similarity. And so that's one of the things that's very important to know, is that when you're assessing something like intra-class correlation coefficient, it's always compared to something else. You can't just say the standard deviation of this is 5, and 5 is high, therefore the ICC is very low, because that doesn't mean anything. In order to have this comparison, you need to have other groups that you can compare it to in order to say that these this variation is in fact too large because it is basically covering the same range as those other groups. And that's a really important idea to have because you will have data sets that have very, very low standard deviations that are still not very similar or don't have this low inter-class correlation coefficient because they're too close to other groups or they're too close to the groups that you're trying to potentially compare it to. And so, on the other hand, you'll have things that have a huge amount of variation, but as long as the groups that you're looking at have even more variation than within a group, you're fine. And so it's really not about the absolute numbers at all, it's about how they compare with one another. This also means that if you're using this to try to decide how reproducible your experiment is, you'll need to have at least one, ideally more, groups that you can compare it to. Because without those groups, you don't have a reference for this number. And so this is one of the very kind of easy tools for saying this value is reproducible enough in order to use it in an experiment. Because if you start to see that the range of this is huge and covers everything else, then it probably doesn't make sense to try to do another analysis with it. So another thing you could do here is if you were focused strictly on reproducibility and you had the same flower, you might have each one of these groups being a different flower, and these measurements being you going back to the sample and measuring again 5 different times what the sepal width is. Because each time you measure it, you probably look at your ruler a little bit differently. You probably put it at a slightly different part of the leaf. And so if you want that to be reproducible, you need to make sure you've done that measurement a number of different times and you're comparing flower A to flower B. And they don't even need to be different species. You just need to show that that value is smaller than the variation between the groups that you're looking at. And so this is for sort of giving you a cutoff. And the way we then define this is by looking at sort of this variation among the groups or classes compared to the variation within the groups or classes. And so this is a very simple formula. There's tools in Python and R and everything else which are able to calculate this easily for you. And basically, 1 means 100% of the variance is between classes, and 0 means that 0% of the variance is between classes. And so these are 2 cases that we probably never actually realize, but they're the 2 boundaries for this value. And so if we actually look at this coefficient for these systems, this one has a ICC of 0.49, which indicate it's not very reproducible. It probably wouldn't be a very good variable to use for further parts of the study. Well, this one has one of 0.96, which indicates it's fairly reproducible. We still see that between these groups, there's probably some overlap, which is why it's not 100%, but they're different enough that it's probably usable. We can do the same sort of thing for coin flips, where you can look at this, where you have 2 coins that you're trying to compare, and you're trying to see how reproducible the flipping is. And of course, with ICC, it's a very poor indicator for a bias in a coin, unless the bias is very, very strong. And so we'll need other tools for actually starting to look at that. Yeah? Yeah, it's comparing all 3. Yeah, so if you just compared these 2, it would probably be 0.9 or 0.85 or something like that.
Would you take the lowest number, or would you take because what would happen if you got this group way off as well, and you got another group way off, and this number would be 0.98?
Right, yeah, then it would be even better. And so you'd want to make sure that whatever groups you were looking at here were somehow representative of the types of experiments that you were doing. But particularly because this is so tied with the variance, if you were doing an experiment, for example, where you were trying to measure the length of things accurately, and the three groups you had were ants, beetles, and whales, you would have a very, very, very high ICC. But that doesn't mean you could necessarily measure the difference between ants and beetles very well. It just means that whales are way, way, way larger than ants or beetles. And so you'd want to make sure if you saw something where these differences were very sort of wildly different, that you made sure that that was representative of the groups you were actually looking at. If you were trying to, it really depends on what you're trying to do. If you're trying to make a tool that classifies it into different groups, then you would kind of say, my goal is to be able to classify accurately into the three different groups. I will include all those different values because I want to have the ICC for the overall system. If you want to say, I'm trying to classify between versicolor and virginica very accurately, and that's what's really important for me, then you would focus on just those two groups. Potentially differentiating these two groups will be the trickier problem to solve, but you're just interested in sort of how similar or how reproducible are the values that you're measuring. And so that's what this is trying to show, much more than the explicit focus on can we tell the difference between group A and group B. So we have other tools for looking at that. And so I guess that's what's, this metric is just sort of the starting point for saying that your measurements are reproducible before you get to how do you actually go about making a classification, making a prediction, grouping these things together, because that's kind of a different task. And so this you would use to say this coefficient or this result is very reproducible. Because it's reproducible, we will then use it for this kind of classification task. So this is where we start to get to the testing aspect. And so, once reproducibility has been measured and assessed, and you're confident in your ability to come up with the same result from the same system, you'll want to start comparing groups. And so the idea here is that you want to be able to say, what's the likelihood that these true groups are kind of different, given the data that you have? And so, who has heard of a t-test before? Okay. Does anyone feel comfortable enough to explain it? Or at least explain what they think it means or what the idea behind a t-test is? It's a function in Excel. T-test, and it will spit out a p-value. Every single scientific paper you read, with the exception of a very few, has a p-value or a t-test somewhere in it. So it's looking at their variances. That's more of the actual kind of implementation details. But what is a t-test actually trying to do? Yeah, so that's what people use it for. So when you look at a paper, people are using a t-test to show if there's a difference between two samples. But that's not, in fact, what a t-test does. So what a t-test does, and this is a very important distinction, is it says, what is the likelihood that these two groups are taken from the same sample, or sampled from the same pool or distribution? And so it doesn't tell you that there's a difference. It doesn't necessarily even tell you how likely it is there's a difference. What it tells you is how unlikely it is that they're sampled from the same distribution. because that's something that's much easier to show. Because what you have with the t-test is basically this very simple statistical model that you have some distribution that you don't know about. It has a mean and a standard deviation, and you have two different things which sample from it in some fairly random manner. And what the t-test says is, how likely is it that these two things were sampling from the same distribution? And so what it kind of intrinsically does is it says, based on the samples that we have, what is the likely mean and standard deviation of the distribution this was looking at? And are these two distributions close enough to be considered similar? So you kind of have a number of little steps inside it. And so you take for each collection of numbers you have, you try to estimate what the mean and the standard deviation are for a distribution, and some sort of confidence interval, because if you have 5 samples, it's very difficult to establish mean and standard deviation for a bigger distribution. And then you take those two groups and look at how different they are and try to assess how likely it is they were sampled from the same set. And so people often say, we used a t-test and the value is less than 0.05, therefore the groups are different. And this is statistically very wrong. You're not allowed to make this statement. All you can say is that the likelihood that they're the same, or they were sampled from the same distribution, is fairly low, or is below 0.05, assuming the distribution was normal and all of the other things about our experimentation and there was no sort of consistent bias or anything like that. And so this is where it's very important to kind of have an idea of what your assumptions are. What are you assuming in your model? What do you know about your distribution? Is there any reason to think that it's normal? I mean, as we saw with the sort of financial crisis, assuming a distribution is normal that is not normal can lead to massive problems when you're trying to make statistical inferences about how this distribution behaves. If you have a distribution that's sort of log normal or power law rather than normal, you will end up with much fatter tails than you would expect, and your ability to estimate the standard deviation from 5 samples is significantly lower. The next step is, once you've established these assumptions, you need to come up with a null hypothesis. So is anyone familiar with this term? So the null hypothesis is really when you assume your default assumption is that they are sampled from the same distribution. So you have, 5 bone mineral densities from mice with this gene on and this gene off. Your null hypothesis is that they came from exactly the same group. And so effectively, your null hypothesis is that you expect no difference between the groups at all. And then you calculate the probability of the observation, so of the measurements that you actually took, given the truth of the null hypothesis. And so if we go back to kind of the coin flip example, this is, we assume the coin is unbiased. If we get 10 heads in a row, how likely is it that this assumption is still true? And so this requires, of course, knowledge about the probability distribution of the data, which is why we always assume, or almost always assume, it's normal. And this modeling can be very, very complicated, which is why this t-test is used so often, because it's a very easy tool. It's a function in Excel, you can just apply it and you get some number out, which makes it look like you know something about statistics. And so if we go back to the simple loaded coin example, what are the number of flips? Sorry, what is the probability that this number of flips are all heads, given that the null hypothesis is true? And so here, if you have one flip, the probability that it's heads, or the probability that you measure all heads, given that the null hypothesis is true, is 50%. I mean, that makes sense. That fits with our expectations. But if you get 5 heads, the probability that the null hypothesis, the probability of getting 5 heads in a row, which is what you measured, with the null hypothesis being true, is now 3%. And so at this point, you are at the boundary of statistical significance. So normally the p-value less than 0.05 is what is used in biology and medicine very frequently to show significance. And we see here that this is at 3.1%. And so getting 5 heads in a row is already, for most journals, a statistically significant result. And for any of you who flipped coins more than 5 times in a row before, you know that getting 5 heads in a row isn't particularly difficult. And so if you think about the reality of all the people doing PhDs or projects, if you spend all day doing experiments, the likelihood that you'll get 5 heads in a row at some point is very, very high. And so it's actually not particularly surprising at all that a lot of these studies are then found later to be kind of irrelevant or not as significant as people thought, because getting 5 heads in a row doesn't mean you have a loaded coin. It just means you were flipping a lot. Getting 10 heads in a row is then slightly more impressive, and that's 0.1%, but it's still not good. And so the question is kind of really how good is good enough, and where do we draw these boundaries, and how do we make sure we're not getting a lot of these results that are meaningless? How can we be more confident in that? And so one of the very interesting things that's happened, of course, with all the automation and scalability that are possible now, and that's one of the things the high content screening lecture at the end will go over in much more detail, is that getting these significant results has become much, much, much easier. If you have to flip a coin manually yourself, maybe you'll find a couple, 5 or 10 significant results in a day. But if you're able to build robots and all the robots do all day long is flip coins, you can then get to these significant results very easily. And so a lot of these tools aren't necessarily very well matched, at least in how they're currently used, to the data generation and automation that are available today. And so when you see this talk that we have in 2-3 weeks, he'll go over a lot of those things, and that they actually don't use things like p-value particularly often, because they're doing millions of experiments completely automated on hundreds of different machines with hundreds of different analyses running. And if they were to get excited every time they had a p-value less than 0.05, they would get excited, every couple of seconds. And so they need something more discerning than a simple student t-test in order to know that something's actually important. And so this is where we have this idea of the student t distribution. This is kind of making a lot of very strong assumptions about your system. So you assume that it's normal, which allows you to then predict fairly accurately without a huge number of observations what that distribution might look like. You can then use a student t-test to kind of get the differences out and get these p-values out. There's links to this. I would recommend reading in more detail how this works if you're interested in it. But this is just one of the very simple models that we have. So one of the most important things to understand with this, which is also then very strongly related to this kind of high content screening, is this p-value of 0.05 is sort of the cutoff. We saw that already 5 heads in a row, you have 3% chance of getting that. And so that would be a statistically significant result. And here is where everything becomes much more problematic. And so if it's just you flipping coins, the likelihood that you get 5 heads in a row is 3%. But if you now have 10 people flipping coins, that goes up to 27%. If you have 20, 40, and if you get to 80 people, so if you think about these large automated experiments, the likelihood is 92% that someone finds something significant. And so quite clearly, that cutoff for a p-value doesn't really make sense if you're having 80 things being done independently in parallel, because we know none of these coins are statistically significant. We are taking completely normal coins. And so when we find that random experiments are giving us significant results, clearly our cutoff for significance isn't good enough. And so, this is also, again, the issue with performing 10 times as many flips. And so many of the methods for doing this involve sort of rescaling that data. And so if you're performing, 80 different people are performing the same experiment, then you set a much higher p-value in order to show that this is in fact significant. And so so this is the same idea as when you're looking at multiple variables or multiple different measures that you're measuring in a study. And so this is actually quite bad news for us, because we've spent most of this lecture coming up with new metrics that are very interesting and very exciting. And so if you've been following all of the lectures and implementing everything, we now have the ability to calculate at least 40 or 50 different metrics for every sample we get. And we initially thought this was a very good thing, because now you can produce a huge amount of data, you have giant tables, you can make all kinds of charts, your bosses are very happy. But we now see that the number of significant results we find is pretty strongly correlated with the number of variables in our study. And so this is with all random variables. So none of them should be significant. They're generated by a random number generator. But here we see if we have 60 variables in our study, on average we will find 3 that are significant, even though none of them are in fact significant. And so this kind of is alarming for us, because you don't want to, maybe if you're trying to finish your PhD quickly and you just want to have something to publish, this is very exciting. But if you're actually concerned about producing meaningful, accurate work, this should be very alarming, because you don't want to do this analysis and find out that you discovered 5 things were significant and you spent years of your life writing a paper that was based on a completely wrong understanding of statistics or what these results mean, which is how I would guess many of you feel. And so this is why we have sort of this multiple testing correction, which is one of the very small things you can do for it, where you can sort of start to divide by the number of tests performed, so that even when you go up to 60 variables, the number of significant findings stays low. And so that's obviously a very simple approach to take. This is called Bonferroni correction, and sort of is normally what happens when you're starting to do a lot of these different tests. And so that seems like it's a fairly good solution. You're now not finding erroneous results anymore. But what might be a problem with that? It's an idea we touched on briefly earlier with the ICC. Yeah. Yeah, so this is always, or almost always, with 2 groups that you're looking at. So you're just trying to show the difference between 2 groups. And that if you have 3, you can do p-tests sometimes, and that will show you, that there's a difference between those 3 groups, but it won't show you what the difference between all 3 of them is. But in terms of the correction itself, dividing your p-value by, the number of variables you check seems to prevent you from from finding these erroneous results. Does anyone see a problem with doing this? I mean, is that what you should do for every study? Just calculate all the variables possible? I mean, we've got, I think one of the companies out there, HealthMine, has a tool for clinicians that will calculate 146,000 metrics for each patient. Should we just do that and then throw it all in a statistical program and see what comes up as significant? Did you have a...
Yeah, just that you were overcorrecting if you have too many variables.
Yeah, so that's exactly what it is. So when we divide this p significance value by the number of variables we check, we then correct for this problem of finding significance where we don't want to find it. The problem we have, though, is that if we have a variable that is in fact different, and let's say there's a 10% difference between the variables between two groups. And so again, this is all made with artificial data, because then we can just use random number generators and see what comes out rather than with real experiments. But if we have two groups and one group is 10% different than the other group, we want to be able to see that. We don't want to find all these random fluctuations that come up, but we want to actually see that these two groups are different. And so what we have here is when we're doing this correction, what p-value do we get for a 10% difference? And so as the number of samples in the study goes up, this p-value goes down. Is that? Sorry, this is a better graph of that. So this is the number of samples in the study. This is the probability of finding a significant variable, given that we have a significant variable in there that has a 10% difference. And so here we can see that if we have one variable and we have 25 samples, we have over a 50% chance of finding that statistically significant difference in that variable. And so that means when we do the student t-test on these two groups, which are randomly generated, we have more than a 50% chance of actually seeing that these two groups that are 10% different are in fact different, which is good. I mean, that's what we want to have. What we see is that if we now have 100 variables in our study, that with that same group of 25 patients, the likelihood that we actually find the statistically significant variable goes from 55% down to 2%. And so this should be quite alarming, because a 10% difference in your group is already quite large. I mean, not huge, but large enough. And this means if you're looking at 100 different variables, you probably won't see that 10% difference anymore with only 25 samples. And in order to get to that same level as we had here, you'll need to measure more than twice as many samples. And so in some sense, it's good, because you want to have bigger studies, and then you can look at more variables and look at more sort of interesting analyses. But if you knew beforehand that most of these 100 variables were garbage and not really very interesting, then you made your study unnecessarily twice as difficult or half as sensitive by adding those 100 variables in. Is that clear? Because this idea is quite important. Because we'll see, I mean, particularly with tools like 9 or Python, you have so many libraries which automate everything, which are capable of producing thousands of different metrics. You can include the entire histogram. You can include histogram of oriented gradients for your image. You can include, thousands of texture features with each analysis you do. Getting to 100 isn't difficult at all. But that's not really the goal. Or if you get to 100, then you need to make sure your samples get way higher as well. Because a lot of the things that we're looking at are very small differences. If you were to find a drug that made people 10% taller, that would be a huge result. Most likely, a really, really effective drug might make someone 1% taller. And so the size of the study you would need, if you were measuring 100 different variables and you made someone 1% taller, would be tens of thousands of people in order to do that well. Yeah.
These four variables that are somehow related, like say I got my height and my weight, but if you measure maybe my height and my color of my jumper, those aren't related, so that's still going in.
Yeah, it still goes in.
The color of my jumper doesn't affect my height.
It doesn't affect your height.
Two tests then?
So what you have here, so this is the sort of very important core concept here, is when you do these multiple tests, you have the ethical responsibility of keeping track of how many of these tests you've already done. Because the more tests you do, the more likely you are to find a statistically significant variable. And so what we saw here was, as you increase the number of variables in the study, and all of these variables are independent, they're all randomly generated, and they're all identical. None of these variables are statistically significant or are actually different at all. They all have exactly the same mean, they all have exactly the same standard deviation. What this graph just shows us is that the likelihood of us finding one of these results goes up as we add more different things we look at. And so even though height and the color of your sweatshirt are completely uncorrelated, the fact you looked at both of them makes the hurdle for finding a difference higher. I mean, that sort of, if you imagine, you could go through someone's head and count all of their hairs, and certainly one of their hairs is probably very correlated with, at what age they're going to die at, or something like this. But that doesn't mean that you've actually discovered something. That just means you looked at a lot of hairs and eventually you found something. And so we have this sort of requirement of, when you start to look at more and more variables, you need to correct for the fact that you're doing these multiple tests. And the way you correct for this is by sort of dragging this line down, which is dividing sort of by the number of different tests you've done. This Bonferroni correction is a very, very simple one. But that means that when you're doing these kind of experiments, if you have a data set and you do, okay, let's look at height, that counts as 1. So, maybe, sweater color's interesting, 2. Maybe eye color, 3. And that you need to be keeping track of this in your head and somehow prioritizing what things you're looking at. Because each time you look at a new variable, you need to be correcting for the fact that you've already looked at 5, 10, 20, 100 different variables. Because if you don't correct for that, by the time you get to 60, you'll have already found 3 variables, even if none of them are interesting or actually different. Yeah?
I have a question in terms of implementing this. I divide by number of samples the p-value I take.
Yeah. I mean, this is just one of the very simple approaches. There's lots of other approaches that you can follow. This is just kind of the general idea. But it's very non-intuitive because it doesn't seem like the fact that you've already done a test for height should affect a test you're now doing for sweatshirt color. That you see those tests as being completely independent. And if you do a big statistical analysis, you check 100 variables, none of them are interesting, so you kind of ignore it, and then your boss comes back 2 weeks later and says, actually, you should look at the inverse entropy of the image. And you do that, and then you find that the p-value is 0.05, and you say, perfect, we can publish now. But you can't, because you've already looked at 100 variables before. And none of them were relevant, and by looking at another one, you have to now correct that by the fact you've already looked at 100. And had your boss come to you initially and said, just look at that inverse entropy or whatever this metric is, that would have then been statistically significant. So that kind of the order you do things in does matter. Which is, I agree, quite counterintuitive, but it means that when you're doing these studies, you want to make sure you're not looking at 100 variables that are completely irrelevant. You sort of pick your statistical tests, and you know that each one you do has a cost. And it makes you less sensitive to small differences. Yeah? Because what we have here, this idea of statistical significance is really kind of how likely is it that you would have stumbled across this result if everything were completely random. And so if you've checked 100 variables and none of them were statistically significant, and you check the 101th and it is, then you have to account for the fact that you've already checked 100. Versus if you had just checked one variable at the very beginning that was statistically, and you found it was statistically significant, and then not checked anything else after that, that would be okay. But sort of the order implies you knew beforehand that this variable should be interesting, and that's why you investigated it early, and so your threshold is a little bit lower. Obviously, whenever you're at the 0.05 boundary, you're really at the cutoff for what things are rare enough to be considered sort of interesting. But even sort of between 0.01 and 0.05, there is lots of, the order does matter. How you handle looking at the variable, if you suspected this to be statistically significantly different from the beginning, and that's what experiment you did, and this is what test you do, it's then a slightly lower burden than if you're just sort of shotgun testing 100 different metrics without any idea, hoping to find one that's statistically different. And that sort of the way for understanding it or getting comfortable with it is just by simulating data. It's very, very clear when you simulate hundreds and hundreds of experiments with completely random data that you will find p-values below 0.05. And this statistical significance, the whole reason we do t-tests, the whole reason all of this exists, is to try to make that unlikely. But it's still going to be possible. And the reason why you have to kind of count each one of these tests you do is to keep it unlikely. That you don't want to have some sort of analysis you're doing where, your data would have given you differences anyways. And so, one of the things that is very easy to do for this, it's not completely a cover, but it might help you, is you can replace all of your data with random number generators. And then you can perform exactly the same statistical analysis, and you can see, did you really find variables that were statistically significant or not? And, most of the time, and you can actually, particularly if you're dealing with student projects, give your students first a set of random data and have them work really hard to try to find differences, because they'll probably find something. And then you've kind of seen, are they actually doing a clean analysis of the data, or are they just trying really hard to find something that's different? But yeah, it is a major problem in science, and there aren't really easy solutions for it, particularly when it is so easy to measure hundreds and hundreds of thousands of variables. Because if someone comes to me and says, I looked at inverse entropy and I just knew it was going to be interesting, and that's the only variable I looked at, this is really significant, I sort of have to believe them. You know, if they're like, I looked at the third coefficient of inverse entropy in this specific analysis, I find it rather unlikely that that was the first variable they chose to analyze. But you kind of have to trust that it was. And so what most people do is they do hundreds and hundreds of different variables, they find one that's interesting, and then pretend like that's the only one they ever looked at, which is then very misleading for everyone else. It's often also the case that you have someone do your statistical analysis who's not involved in the study at all. So if you look at a lot of medical studies, they have a professional statistician who gets the data completely blinded of the experiment, who doesn't care about the results at all, who's just being paid per hour to kind of go through the tables, versus the person who did the experiment, who spent 2 years of their life trying to, get this result, who can't, get promoted to the next level if they're not able to publish it in an exciting journal. That you want to disentangle those things as much as possible. Yeah.
So, if I understand you correctly, it means if there's no physical underlying reason why it is the third coefficient in the inverse, whatever, then this test doesn't say anything if that's not the first thing you looked at. But if you look at 100 different properties, compare them, find nothing, and then you look at the third inverse, and you also figure out that it is an underlying physical reason why it is that, then the significance would be restored, because then the report, it doesn't matter, right?
This
is only relevant if you don't have an explanation for the correlation, and you're just looking randomly for a correlation between the properties.
No, it's relevant for both, because, as you will see, any professor at ETH will be able to come up with an explanation for why this variable is significant. It doesn't require that much creativity. You can go very easily to your boss, show them a result, and say, look, there's a statistically significant difference there. And they'll say, oh yeah, it's because of this and this. And then go them to the next day with the opposite result. And they'll say, oh yeah, that makes sense too.
Okay, but if you have the prediction, the physical prediction why it is, and you have it beforehand, put it in a drawer, you check 100 values, and then you check that one that predicted, and it has a significance.
But you shouldn't do it like that. If you had a physical prediction and you knew this variable was going to be important, why did you test the 100 before? You should have just tested that one first.
Okay.
And that when you start to do this kind of batch scale statistic where you're testing hundreds of things, you make it more difficult to find these small differences. And so that's why you really want to be very sort of prudent with the analyses that you do.
So it's like what came first, chicken or egg kind of thing.
Yeah.
If you knew that it has to be there and you find it, then it's proving more than if you say, I found there is a correlation and I estimated a reason why that is.
Yeah. That's sort of the underlying idea, is that if you have some reason to be, because you will always find an explanation for whatever results you get. There's always something of, oh yeah, blue people are taller because there's a lot of Swedish people who are tall. You can come up with any explanation for any result you'll ever get. Being able to come up with an explanation afterwards does not mean that there's actually a link. And so this is about being very careful from the beginning in how you go about this. And this is probably one of the most important things you do, because, as you've seen from economists, from these, there is a huge problem in science with reproducibility and statistical significance. And so at least be very, very aware of how you're supposed to work and how things are supposed to be done. If that's, in fact, how it's done in your lab is something I can't attest to, but if you at least know how it's supposed to be done, you're less likely to make mistakes there. Yeah?
When you null hypothesis, you say from the start, instead of going and mining the data, you go say that this sweater color influences the height.
Right.
And then you test that, and you just have to say,
No.
Isn't this now you have got an hypothesis, you're just looking for something?
Yeah, but even if you're looking for, 100 different things, you have to,
Use this correction.
yeah. But even if you're looking at 10 different things, you need to use that correction. Even if you think, it's probably changing sweater color and weight and height and eye color or something else, you have to correct for the fact that you're doing all of those tests. Or that, your boss will say something like it'll change shape. And well, we have, 65 different metrics you can use to measure shape. So if you think it changes shape, then you'll look at all 65 different of those metrics, and one of them might be significant, but you need to be correcting for the fact that you looked at 65 different things.
In that same run.
Yeah.
That same model.
Yeah. Boom. Okay, so it's 11:11 now. So I guess we will move the predicting and validating to the next lecture. Otherwise, we won't have too much time to get over this lecture stuff, the exercises and things. But yeah, I think this is important enough that it's worth emphasizing very well, because this is kind of the core of experimental science or theoretical science. I mean, it's the core of science. So yeah. So for the exercises, they're available in KNIME as before, and we have some Kaggle workflows where you can look at these kinds of analyses. There's also the IRIS data set. I mean, it's a very small data set. You could have it on your laptop as well in Kaggle. And the interesting part of this here is that you have lots of nice kernels that have already been made for visualizing and looking at the tools. And so you can play around with the data sets quite well. So like this one is quite cool for looking at sort of decision trees and boundaries. This is a fork of the original one. Where you can then start to see how does this data look and what sort of other things can you do with it. So yeah, here you see kind of those different flower groups that we had before and kind of what approaches you can take for looking at it and visualizing it. We also have in our own exercises I think here, if they've been updated. Some tools in R. There's obviously Python equivalents of these tools as well, but one of the nice ones is called compare groups, which then allows you to put in a table and say what variables you're interested in, and it will then come up with a nice summary of what you're looking at. And so you can see then what groups are statistically significant and what the differences are, and then start to plot the sort of residuals and things that come out of these models. Yeah, the prediction thing we'll get into next week, but that covers it more or less for today. And then very similar exercises are available in KNIME for those of you who are using that tool. So are there any questions on this? Okay. Good. So